KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Free bus for LKG to 8th standard.
Free, hygienic and nutritious food.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Okay, man. Now, in previous video, what is information we have seen now? In processes of recombinant DNA technology. So, we have seen different types of side headings. So, let me revise those. So, first one is isolation of genetic material we have seen. Don't forget about first one is isolation of genetic material we seen. Next one second side heading is cutting of DNA at specific locations we seen. Next one there after completion of cutting of DNA at specific locations. So amplification of gene of interest using PCR. So we learn. Next one insertion of. So insertion of recombinant DNA. So means here this point we need to learn. So insertion of recombinant DNA into the host means in previous video means what are the side headings we have seen. Isolation of DNA. So isolation of DNA we have seen. Next one cutting of DNA at specific locations we have seen. Next one amplification of desirable gene with PCR we seen. Now according to the textbook, actually this side heading is not there. Now we are seeing now ligation of once you see there ligation of a desirable or a desired DNA fragment with a suitable vector. So actually this side heading is not there. So by referring another material, so giving this information to you. So from here onwards, now let us have a look. Now ligation of the desired DNA fragment with the suitable vector. Now when we will be obtaining, so or when we will obtain which one, what we will obtain desirable DNA fragment. So that desirable DNA fragment now we are going to ligate. Ligate means join. With whom we are joining? With suitable vector. So did you understand one point? So we are adding desirable DNA fragment with suitable vector. So automatically what we can prepare? Once you come to the last line, so here you will find the answer recombinant DNA. So definitely we can prepare or we can construct the recombinant DNA. Point you understood or not? So already I have given so many times recombinant DNA is nothing but the combination of two different types of genes together. So means here once if you get a desirable DNA fragment, that particular desirable DNA fragment, definitely we will incorporate or we can incorporate into the suitable vector. So definitely at the time now we can see the formation of recombinant DNA. So that now we will let us see the information. Now the desired DNA fragment means which one? Gene of interest. Desired DNA fragment, that is a gene of interest from the source DNA. From where? From the source DNA means from where we have obtained, that we can be taken as a source DNA and cut the vector or mixed. Means here the vector or mixed and ligase is added. Means here from the source of DNA we are getting desirable DNA fragment and vector also here we are getting that vector as well as so DNA fragments. So by using of same restriction enzyme we are cutting and now here the both are mixed together. Mixed together, how these two are mixed together? With the help of DNA ligase enzyme. So definitely which one formation takes place means recombinant DNA formation takes place. Don't forget. Means in this particular case, what is the point here? Mainly we have to understand means for the cutting of DNA fragments or for obtaining of gene of interest. So and for the obtaining of or for the taking of which one vector. So these two things means DNA source as well as vector or the plasmid, we have to cut with the same kind of restriction enzyme. So then only we can get same kind of sticky ends. Already in previous classes I have given now, so that is the information. Now, so finally, now what we have obtained? Recombinant DNA we have obtained. Now come on to the next side heading, that is insertion of recombinant DNA into the host cell or the organism. One more time, insertion of recombinant DNA. Insertion of recombinant DNA into the host cell or organism. So in this particular case, no, so we can discuss regarding competent host. So in yesterday's class I have given no competent host. So means, sorry, here the competent host why we are preparing means we are going to study the insertion of recombinant DNA into the host. Definitely. So what is a desirable gene is there with the vector, no, in that particular case, no, that particular combination of two genes we will be calling as recombinant DNA, no. So that particular recombinant DNA now we have to incorporate into the host cell. So how we can insert into the host cell? So there are yesterday's class concerned, we have seen different types of techniques, no. So by using of different types of techniques, so definitely we can incorporate. So means here what is the point? So first of all, one important method we see, no. So what is that? So first of all, bacterial cells definitely we have to treat with the divalent cation. Which one? Calcium also we have taken, no. At the time small pores formation takes place on the holes of on the wall of bacterial cell wall. At the time, no, the recombinant DNA as well as a host cells we have to place on the ice. Later at 42 degrees centigrade we have to give heat shock treatment, and later ice treatment when we will be giving, no, at the time definitely this particular host receives the recombinant DNA. Point you understood or not? So that is information we have seen under competent host. And next one, in animals most suitable method is microinjection, and in plants most suitable method is gene gun method or biolistic method. So we have seen. Next one, and last and final technique, that is disarmed pathogen also we have seen. Actually under competent host, so why I have revised all these things means, if I revise all these things, so chance is there to recollect. So that's why I have revised. Try to revise all these things by one more time. So that is the information now. So there are several methods. So just here name only, just one words only they have mentioned. There are several methods of introducing of ligated DNA into the recipient cells. So means here just simply means what are the methods we have been discussed till now, no. So regarding that one, just they have mentioned just several methods are there for introducing of our recombinant DNA into the host cells they have given. Recipient cells means host cells, the means where we are going to add or where we are going to insert our foreign gene so into the particular cells. So those kind of cells we will be calling as recipient cells or host cells. And understood now? Now let us move forward. Now these recipient cells after making them competent, so now here we have to make them as competent to receive the so here next line also they have given like that only, to receive take up of or take up DNA present in its surrounding. So definitely when we will prepare which one competent host, so to receive the which one DNA present in its surroundings, so we have to make it ready. So then only that particular host can receive the recombinant DNA. So if a recombinant DNA bearing genes for the resistant to the antibiotic, so that is ampicillin. Here ampicillin completely I didn't write, just AMP I have written. AMP indicates ampicillin, you try to understand like that. Now here what is the thing? Suppose if the recombinant cells, so where it is, once you see there. So if a recombinant DNA bearing a gene for resistance to antibiotic, that is ampicillin, is transferred into the E. coli cells, the host cells become transformed into the ampicillin resistant host cells or ampicillin resistant cells. So in IPO what is information now we are learning now. Once you see recombinant DNA is there. So that particular recombinant DNA now it is there outside of the host. Now that particular, suppose our recombinant DNA is there, no, means with a vector our desirable gene is there, no, suppose our desirable gene has ampicillin antibiotic resistance. So this particular, when we will be incorporating this recombinant DNA into the host cells, no, so because of the presence of our desirable gene, because of the presence of our desirable gene, now host cells also getting antibiotic nature. Emma, did you understand? So 1 minute, one more time I will explain, just you listen, concentrate on my voice only, Emma, don't see the board, just you listen. So what is the information now I am going to take now? Emma, one desirable gene we have in our hands. Now that one we are keeping into the plasmid, isn't it? So now what we have obtained, recombinant DNA we obtained. Now on our desirable gene, Emma, now that particular desirable gene has antibiotic resistance. Which one? Ampicillin. When we are going to introduce this recombinant gene, means recombinant gene is nothing but desirable gene with plasmid. So that particular thing will be there in E. coli cells. So now because of the presence of our foreign gene in E. coli cells, so E. coli cells also now obtaining the capability to resist the ampicillin, means definitely that particular cells can grow in ampicillin containing medium. Am a point you understood or not? Ampicillin containing medium. So why? Because means here the host cell getting ampicillin resistance because of the presence of our desirable gene. Why? Because the desirable gene has the capability to resist against the ampicillin. Am a point you understood or not? So try to understand this one. If you spread the transformed cells on agar plates containing ampicillin, so here what is the thing? Only transformants will grow, means so now these particular cells, host cells, we will be taking as, or E. coli cells, here we have taken example, no. So these particular transformed cells, suppose if you are growing on agar, so that particular agar containing ampicillin antibiotic. So because of the presence of antibiotic nature, where in a host cell, definitely it can be grown in ampicillin containing medium. Ampicillin containing medium only. So definitely this particular host cell will grow. Why? Because particular host cell is obtaining antibiotic resistance because of the presence of foreign gene within it. So that's why here the host cell also can grow in the ampicillin containing medium. Okay, I hope you understood this one. Next one, only transformants will grow. Means transformants means one bacteria or a host, within the host recombinant DNA. So such kind of organisms only will grow. Other remaining things means who is that? Untransformed recipient cells. Untransformed recipient cells will die. So untransformed means, so host will be there, but there our desirable gene will not be there. Desirable gene suppose is there, means so that particular host also will obtain resistance against the ampicillin, but here untransformed recipients will die. Why? Because due to the absence of recombinant DNA. So try to understand clearly. So now, since due to the ampicillin resistant gene, one is able to select a transformed cell in the presence of ampicillin. So thereby, so over here what is the thing we can be seen? Since due to the ampicillin resistant gene, so means here due to the ampicillin resistant gene, so one is able to select a transformed cell into the presence of ampicillin, or transformed cell in the presence of ampicillin. So till now that explanation only I have given. Next one, the ampicillin resistant gene in this case is known as selectable marker. Means this particular ampicillin resistant gene means what is the gene we have been incorporated into the plasmid, and what is the recombinant gene is present in the host, because of that particular foreign gene. So that particular entire host are now getting resistance against the ampicillin. That's why this particular ampicillin we will be taking as selectable marker. Don't forget selectable marker. Now obtaining foreign gene product. Obtaining foreign gene product. Now this explanation also is important. In obtaining of a foreign gene product, before this explanation, before read and explain this one, now orally somewhat information I will give you. Now one foreign gene, okay, obtain foreign gene product. obtain foreign gene product. Ipo in a molecular basis of inheritance chapter I have given some more information. What is that? Suppose particular DNA is there, means that DNA undergo the process of transcription, later undergo the process of translation. Likewise I said no, means by the process of transcription, so mRNA formation takes place from the DNA. Next one, this particular mRNA undergo the process of translation, so there proteins formation takes place, I said. So likewise same concept only now we will discuss here. What is that? Obtaining the foreign gene product. Obtaining foreign gene product, how we can obtain foreign product? So definitely we can obtain from the foreign gene. So here this particular gene undergo the process of transcription, so thereby mRNA formation takes place. Later by the process of translation, that particular mRNA sequence later translated into proteins. Means, so why we are incorporating our foreign gene into the host cells, means for obtaining of desirable product. So definitely we can obtain a desirable product when we will incorporate desirable gene into the host. So try to understand. It is actually highly concept, means this one we cannot understand by at a time, but unfortunately we are teaching like that, you try to understand. Otherwise, suppose if you are getting any doubts here, one more time I will explain in presence of you. Okay, so now let me continue. That is this, when you insert a piece of alien DNA into the cloning vector and transfer it into the bacterial or plant or animal cells, the alien DNA get multiplied. So in this particular case, just we are inserting a piece of DNA or piece of alien DNA into the cloning vector, and after completion of after creation of recombinant DNA, so that one we are incorporating into the bacterial cells, or we can keep into the plant cells, or we can keep into the animal cells. Means here, suppose if you are doing practicals bacteria related, no, so that particular recombinant DNA you have to place in bacteria. Suppose if you are doing practicals related to the plants, so that particular recombinant DNA you have to place in only in plants, likewise animals also. Next one, the alien DNA gets multiplied. So when we will be incorporating into the host cells, no, so definitely our foreign gene or definitely our alien gene get multiplied. So try to see this one. Next one, in almost all recombinant technologies, the ultimate aim is to produce the desirable protein. Did you understand? Means ultimate means the first and the last, means the completely the last one is ultimate. Ultimate technology and ultimate aim is, what is that ultimate aim? So to produce desirable protein. So definitely we have to produce the desirable protein. Hence there is a need for the recombinant DNA to be expressed. So here definitely so in order to obtaining of a desirable protein, definitely our desirable recombinant DNA must be expressed. Expressed is nothing but, so by the process of transcription as well as translation, so proteins formation takes place. That is only information. Next one, the foreign gene gets expressed so under appropriate conditions. So here foreign gene definitely is expressed in the sense, so definitely with the help of a foreign gene, definitely proteins formation takes place only in case of appropriate conditions. Means when you will provide suitable conditions for this foreign gene and the host, no, so then only that particular host, means where we have inserted our foreign gene, so that particular host will be multiplied along with the host, no, our foreign gene also will be multiplied. Appropriate conditions, definitely foreign gene is get expressed. Next one, the expression of a foreign genes in host cells. The expression of a foreign gene in a host cells involves understanding many technological or technical details. So that information, so means here, so definitely in order to obtaining of or in order to creation of optimum conditions or appropriate conditions, means I think so some other place I have written, so means there are pH and later temperature as well as vitamins, oxygen, water, likewise there are different types of appropriate conditions we need to create. So I have written some other place, so definitely I will give you information regarding that. Next one, after having cloned genes of a gene of one more time, after having cloned the gene of interest and having optimized conditions induce the expression of the target protein, one has to consider producing it on the large scale. So, here definitely when you will provide suitable conditions to the gene of interest, definitely when you will provide optimum conditions, so definitely from that particular gene, so we can see the expression, so thereby we can obtain target protein, target protein. So, that particular protein also we can obtain in large scale. So, large scale means, so when you will provide optimum conditions, so definitely very nice product or very nice yield definitely you can obtain. So, that is the thing they are saying. So, one has to consider, so what we have to consider, producing it in a large scale. Okay, this one we have seen. If any protein encoding gene is expressed in a heterologous host is known as recombinant protein. Means here we are producing one protein. So, this particular protein actually, so from where we are obtaining? From the desirable gene we are obtaining. Actually that particular desirable gene is not exactly belongs to that particular host. So, that one we have brought from the some other place. So, that is why here heterologous host they are saying, means this particular DNA means what is a desirable gene we have taken for in order to obtaining of desirable protein. That DNA is not actually not belongs to the particular or not belongs to this host, that is belongs to the some other host, but here we are obtaining proteins in some other host. So, I hope you understood this. Means here we are changing the DNA sequence and we are getting the DNA sequence from some other organism and we are incorporating into the some other organism. In another organism we are obtaining the protein. So, that particular means where we are obtaining the protein. So, that particular organism here we are taking as heterologous host. So, that particular thing where we are obtaining that particular host only. So, definitely what we can obtain? Recombinant protein we can obtain. Recombinant protein here we can obtain. Next one, the culture. The culture may be used for the extraction the desirable protein and then purifying it by using different separating techniques. So, here there are different types of cultures over there. So, these are different types of cultures. So, definitely we can use for the extraction the desirable protein. So, definitely, so different types of methods or the different type of cultures we can use for the getting of desirable protein. So, then we can get purifying. So, means here definitely if we are producing, if you are getting one protein means, so there are different types of purification methods are there. So, definitely we can obtain purifying, which one purification of a recombinant protein. So, by using of different types of separating techniques. So, likewise some more information we can be seen. Next one, continuation concern. The cells can also be multiplied in a continuous culture system. Once you see there, suppose here the cells can be multiplied, means here when we will produce or when we will create suitable conditions, definitely. So, by using of all optimized condition or the suitable conditions, our foreign gene is multiplied. So, means in this particular case some kind of technique cultures are there. So, we no need to stop any kind of procedure. So, definitely we can maintain or we can do as continuous culture system. Means, so what is the meaning? Here meaning also I have written once you see, that is where in the used medium is drained out. Means here in one system, means in a continuous culture system, from one place definitely we will be so here the medium is drained out, means here unutilized waste materials will be drained out from the one side and refresh medium is added from the another side. Means here suppose here one box is there, means so here two lets will be there. So, two ways will be there. From one way we will add the medium. So, and another way, what is unutilized medium or like a waste material, suppose if anything or there means, so in another way we will remove remaining things. So, in this particular case, so what is the information we are going to take means, So definitely there are two ways are there, means for the addition of a medium as well as for the draining out of a medium. So when we will be doing like that, no, so here we no need to stop any kind of a process. So here we can done the continuous culture system. Continuous culture system. Next one, maintaining the cells in their physiologically most active log exponential phase. So here already in first year studies we learned one point, that is sigmoid growth curve. Did you remember? So there in plant growth and development lesson I have given information, sigmoid growth curve there in exponential phase, no. So there are three stages are there, lag, log and stationary. Likewise we see, no. So likewise here also, here in this particular continuous culture medium, no. So when you will maintain, which one you will maintain most the cells with most physiological activity. So when cells will maintain with most physiological activity, so thereby this type of culturing method produce larger biomass leading to higher yield of desirable or desired proteins. Means here, so when we will be maintaining, so what we will be maintaining, not lag one, just we are maintaining second stage. Second stage is nothing but log stage or exponential stage. There only we can find the more productivity. Okay, so thereby what we can be obtained means, so here we can produce the large scale of biomass leading to the higher yield of desirable protein. So definitely, so this kind of means a very huge, in huge quantities, definitely we can obtain the huge quantities of biomass. So definitely, so thereby we can get high yield of desirable protein. Okay, somewhat information they have given. Next one, the small volume. So small volume cultures cannot yield appreciable conditions, appreciable quantities of products. So one more time let me read. So small volume cultures so cannot yield appreciable quantities of products. Suppose very small minute quantities, suppose if you are maintaining, means here the size of the bioreactors, the size of the so size of the bioreactors is very small. Suppose if you are going to produce which one, proteins or the products in small quantities, so definitely we cannot get very huge quantities of proteins. So appropriately, so what is information, appreciable quantities we may not get. Okay, so that's why to produce large quantities, one more time let me explain, small volume cultures cannot yield, means cannot give which one, appreciable quantities of products. Why? Because we are culturing in small volume. So if you culture in small volumes, how we can get large volumes of products? We cannot know. So that's why what we have to do, to produce in large quantities. So suppose if you would like to get more proteins means, so definitely, so to produce large quantities, so the development of bioreactors where the large volumes means approximately 100 to 1000 liters of culture can be processed and was required. Means here definitely if you would like to get more percentage of yield or more yield means, so definitely you can be switched on to the, or you can use bioreactors. These bioreactors only definitely, so what is the volume we can be, or how many liters of like proteins we can be obtained means, or yield we can be obtained means, so means here the volume of bioreactors is 100 to 1000 liters. Means here this much of a huge stainless steel vessels if you use means, so there, so definitely we can obtain very huge quantities of proteins. So that is information. Next one, thus bioreactors can be thought of a vessel. So already I said, so bioreactors are nothing but vessel like structures. So stainless steel also means here in a microbes in a human welfare chapter, so they have mentioned these bioreactors as stainless steel vessels. So in which raw materials are biologically converted into specific products. Don't forget. So here raw materials means, suppose if you would like to get a suitable product means, or in here we have to obtain, or here we have to produce or here we have to create suitable conditions for the growth of the host. So in that particular case, food materials of that particular host means here suppose related to the particular host, what is the material it will eat as a food, so such kind of materials we will add as raw materials. So these raw materials are biologically converted into specific products. So next one, individual enzymes and etc. Okay. Next, using of microbial plants or animal or human cells. So human cells, a bioreactor provides optimum conditions for the achieving of a desirable product by providing of optimal growth conditions. So here at this point already I told you. So in this particular case, in order to obtaining of which one, if you would like to get any kind of suitable product, so either maybe wherever you want to produce, okay. So definitely, so by using of bioreactors and by using of or by creation of or by providing of optimal conditions, definitely we can obtain desirable product. So how? By providing of optimum growth conditions. So here we need to provide optimum growth conditions. So what are the growth conditions are there? Temperature, pH, substrate, salt and vitamins, oxygen. Likewise there are different types of growth conditions or optimum growth conditions when we will maintain. So definitely, so what is a product you would like to obtain in large quantities? So definitely you can obtain in large quantities, but here, so suitable raw materials or optimum conditions of raw materials you have to provide.